everyone. My name is Jiayi Liu, and here I'm honored to be here to present our recent study, Optimizing Sequence Design Strategies for Perturbation MPRAs, a computational evaluation framework. Our work focuses on the dancing techniques used in perturbation method massively parallel reporter assays, or MPRAs, which are pivotal for understanding the roles of non-coding regulatory elements in gene expression. First, a little bit about the background and motivation. Since the human genome's non-coding regions, including enhancers, plays crucial roles in regulating gene expression, these regions contain binding motifs for transcription factors and are central to various biological processes. However, traditional methods like luciferase reporter assays are limited in throughput, especially when investigating numerous non-coding regions. To address this limitation, MPRAs were developed, allowing us to measure the activity of thousands of regulatory elements in a single experiment. Perturbation-based MPRA take this a step further by assessing the regulatory effects of TF binding motifs. Despite their potential, there's been a lack of comprehensive computational evaluation of different sequence design strategies for these assays. This gap motivated us to develop a framework for evaluating and comparing various perturbation strategies. In specific, we proposed a framework to benchmark 3 different perturbation approaches using a publicly available dataset generated by our team. This dataset includes wild-type sequences, motif perturbation sequences, and negative control sequences. The 3 perturbation approaches we evaluated are PERT1, replacing target motifs with a constant non-motif sequence identified specially. Perturbation 2, similarly to perturbation 1, but minimizing the motifs across the entire genome region. Perturbation 3, which shuffles the nucleotides of target motifs randomly. We defined 5 key metrics to evaluate the perturbation strategies. First, hit rate. It measures the in situ elimination of the target motifs. Second, the perturbation rate, which indicates the impact on target motifs both in situ and ex situ. Third, perturbation specificity, which is the proportion of wild-type motifs that survive the perturbation. Fourth, the newly introduced target motifs per sequence, which identifies unintended new motifs created by perturbation. Fifth, the general alterations in the number of motifs, which assesses the non-specific impact on motif counts. Our analysis revealed some critical insights. First, the perturbation 3 method demonstrated the highest perturbation specificity but introduced the most new motifs, indicating a trade-off between specificity and unintended motif creation. Besides, perturbation 2 and perturbation 1 showed higher hit and perturbation rates, but both of them were prone to introducing systematic biases due to the insertion of fixed non-motif sequences. Finally, across various benchmarking metrics, perturbation 3 consistently showed robustness, particularly in predictive modeling of regulatory activities. In summary, our study provides a comprehensive evaluation framework and benchmarks various perturbation strategies for MPRAs. We recommend using the nucleotide shuffling approach the perturbation 3 for designing sequences in perturbation and MPRA experiments, followed by coherence checks to prevent unintended variation. Our findings offer valuable guidelines for researchers in the field, paving the way for more accurate and robust studies of non-coding regulatory elements. This work not only advances the computational method for MPRAs, but also contributes to the broader understanding of gene regulation mechanisms. Thank you for your attention, and we are happy to take any questions you might have about our study.